if you break a bone, your body basically knows exactly how to fuse it back together.
Right, it's automatic.
Yeah, exactly. Or like if you slice your finger cutting vegetables, the skin scabs, it remodels and it heals. We are so used to thinking of the human body as this, remarkably efficient repair machine.
Which it usually is.
It is. But if you suffer a heart attack, the way your body tries to heal you is actually the very process that ends up destroying the heart.
Yeah, it's a really tragic biological paradox. The rescue effort itself becomes the ultimate cause of failure.
Because the human heart it lacks the intrinsic ability to regenerate muscle tissue after a massive ischemic event.
Right, exactly. Instead of rebuilding that complex synchronized pump, the body's emergency response is to essentially pave over the damage with biological concrete.
Which leaves you with a heart that can't fully beat. And that just leads to a slow decline into heart failure.
And fixing that underlying scar it's always been science fiction.
Until now. Welcome to this deep dive. Today we are looking at a 2026 breakthrough paper from the journal Cell Stem Cell by Wu and colleagues.
It's an incredibly exciting paper.
It really is. Our mission today is to unpack this completely new engineered cell therapy called pCAR-Mos. We aren't talking about a drug that just manages symptoms or, slows down the decline.
No, we are talking about an autonomous living therapy. One that's designed to literally eat the dead scar tissue and force the heart to regrow fresh muscle.
It represents a fundamental shift from symptom management to actual disease reversal.
Yeah, the researchers engineered what they call a bulldozer and builder strategy. But to appreciate how that works, we really need to trace the exact pathology of myocardial infarction.
Which is just the medical term for a heart attack.
Right. So the initial trigger is ischemia. That's a sudden restriction in blood supply, usually from like a blocked coronary artery.
And when that happens, the cells get starved of oxygen.
Exactly. Stripped of oxygen, billions of cardiomyocytes, those are the specialized muscle cells that generate your heartbeat, they rapidly suffocate and undergo necrosis. They die.
And when billions of cells die all at once, they don't just quietly fade away, do they?
Not at all. Their sudden rupture dumps all this cellular debris into the surrounding tissue. And that sets off a massive, just deafening inflammatory alarm across the entire immune system.
Like sirens going off everywhere.
Exactly. And that intense inflammation summons a very specific population of cells called resting cardiac fibroblasts.
Okay, so what do those do normally?
In a healthy heart, they're essentially quiet structural engineers. They just maintain the subtle extracellular matrix that, holds the heart together.
But under the chemical distress of a heart attack,
they undergo radical transformation. They activate into what we call myofibroblasts.
So these are the emergency first responders frantically trying to seal the breach, just so the heart wall doesn't rupture right then and there.
You've got it. They aggressively pump out an incredibly dense extracellular matrix, primarily these heavy collagens.
And that's what forms the fibrotic scar.
Right. It seals the wound, but the physics of this new tissue are entirely wrong for a heart. The scar is stiff, it's rigid, and it's akinetic.
Meaning it can't contract.
Right. You've replaced a flexible beating muscle with a patch of unyielding rebar.
Ouch. And because a portion of the heart is now paralyzed, the remaining healthy muscle has to, pump twice as hard to maintain blood pressure?
Exactly. Which eventually exhausts the organ and leads directly to end-stage heart failure.
Okay, let's just do a quick recap here to make sure we've got the foundation.
Sure, that's a good idea.
So, ischemia kills the heart muscle. That triggers a panic response. The quiet fibroblasts mutate into these active scar-pumping factories called myofibroblasts, and they fill the heart with stiff collagen that ruins its ability to pump.
That is the perfect summary.
So the key to curing heart failure is getting rid of that biological concrete. We have to clear out those overactive myofibroblasts.
But the heart is a dense, critical organ. You can't just, drop a bomb on it.
Right. You need to target the scar builders without damaging the surviving healthy muscle.
And that is where the researchers identified a highly specific target called FAP, fibroblast activation protein.
So FAP is basically like a glowing neon sign worn almost exclusively by the bad cells, the ones actively building the scar.
That's a great analogy. To find that neon sign, the team utilized a technique called single-nucleus RNA sequencing, or snRNA-seq.
I love when we get to talk about snRNA-seq. Explain how they did that here.
Well, rather than just looking at a slice of tissue under a microscope, which is kind of like looking at a blurry crowd of people, This technology allows scientists to isolate tens of thousands of individual cell nuclei.
Wow.
Yeah, and then they read their specific gene expression profiles one by one. It's like checking the ID badge of every single cell in the organ.
So it gives this ultra high definition map of the cellular environment. And when they looked at that map, the FAP protein just stood out.
Incredibly clearly. In a healthy, uninjured heart, FAP expression is essentially zero. It just doesn't exist.
And it doesn't show up in other major organs either, right? Even after a heart attack.
Right, but exactly 7 days post-injury, right when the scar formation is at its absolute most aggressive state, FA2 expression suddenly peaks in the heart.
So they caught the culprits red-handed.
They really did. The sequencing data revealed a direct causal link. The specific fibroblast subsets that express the highest levels of FAP also had the highest ECM scores.
Meaning their RNA was basically screaming the instructions to produce heavy collagens.
Exactly. It's definitive proof that FAP-positive cells are the primary drivers of pathological scarring.
Okay, time for another quick summary, and I want to pose a question to you listening.
Go for it.
We've established the target. The bad scar cells express a unique protein called FAP. So think about this. If you could put a glowing neon sign on any problematic cell in your own body to make it visible to your immune system, what would you choose?
That's a profound question, because once you have the sign, you have to decide how to attack it.
Right, and it makes you wonder, if scientists know exactly what these rogue cells look like, why has it been so hard to hunt them down? I mean, we've had CAR T-cell therapy for years.
Chimeric antigen receptor T cells, yeah.
Right, they cure blood cancers. We can program a T-cell to hunt a specific protein. Why not just program a standard CAR T-cell to go hunt the FAP neon sign?
It seems logical, right? But the physics of the solid organ microenvironment make that incredibly difficult.
That's so.
Well, CAR T cells are exceptionally lethal assassins when they're floating in the bloodstream or the lymphatic system, hunting leukemia. But a heart scar is a dense, physically formidable barrier.
So they can't get in.
When T cells attempt to penetrate that thick collagen matrix, they literally just bounce off. They lack the physical machinery to dive deep into solid fibrotic tissue.
Which means the vehicle carrying the therapy matters just as much as the therapy itself. Okay, so if T cells lack the machinery to dig through the scar, the next logical choice would be macrophages, right? I mean, their entire biological purpose is to infiltrate tissue and phagocytize or eat pathogens and debris.
Yes, macrophages do have the physical capability to burrow into the tissue. The barrier there is biodistribution.
Biodistribution, meaning where they actually end up in the body.
Right. When you inject millions of engineered CAR-macrophages into a patient's bloodstream intravenously, they encounter the mononuclear phagocyte system, particularly in the liver.
Oh, because the liver is essentially a massive biological filter.
Exactly. It's lined with specialized cells called Kupffer cells. These liver cells intercept and sequester the engineered macrophages long before they ever reach the heart.
Oh, wow. So you end up delivering your super expensive, highly engineered therapy straight to the wrong organ.
Yeah, which is obviously a huge problem. T cells bounce off the armor and macrophages get trapped in the liver filter.
This forces a complete rethink of the delivery mechanism, which brings us to the core innovation of this paper, the monocyte.
The monocyte.
I like to think of them as the body's natural circulating ambulances.
That is exactly what they are. They are circulating precursor cells. Unlike T cells or mature macrophages, monocytes natively live in the bloodstream, continuously patrolling.
Just waiting for a call.
Right. And crucially, their surface is covered in specific chemokine receptors. When the heart suffers an infarction and is overwhelmed by inflammation, it releases chemical distress signals into the blood.
The sirens.
The sirens. The monocytes detect these chemical sirens and naturally home directly to the site of severe inflammation.
So they already have a built-in GPS for heart damage.
They do. And once they arrive at the damaged heart tissue and exit the blood vessel, the local microenvironment triggers them to differentiate into macrophages right there on site.
Wait, so they transform into the necessary tissue-eating machines precisely at the coordinates of the scar.
Exactly. Completely bypassing the liver's filtration system.
That is wild. And to prove this ambulance system actually works in a living mammal, the researchers set up this fascinating in vivo tracking experiment.
The IVIS experiment, yes.
Right. So they engineered FAP targeting CAR-monocytes, which they call pCAR-Mos, and they ran them head-to-head against CAR-macrophages.
They tagged both sets of cells with a fluorescent dye called DR.
And injected them into the tail veins of mice.
Yes, and by using an in vivo imaging system, they literally watched the cells travel through the body in real time. The visual data is just stark.
What did they see?
Well, the CAR-monocytes showed a 2.6-fold higher accumulation directly in the infarcted heart tissue compared to the macrophages.
2.6 times higher. That's massive.
It is. And more importantly, they achieved a 4.8-fold higher heart-to-liver ratio.
So the monocytes successfully rode the inflammatory chemokines straight to the heart, almost entirely evading that liver trap we talked about.
Exactly.
There is also a huge logistical advantage here for anyone thinking about how to actually manufacture this drug for thousands of patients, isn't there?
Oh, absolutely. To build a CAR-macrophage, you have to extract precursor cells, put them in a bioreactor, chemically force them to differentiate into macrophages over this long period, and then genetically engineer them.
That sounds slow and expensive.
It really is.
Yeah.
But by engineering the monocytes directly from the bone marrow, scientists skip that entire ex vivo differentiation phase.
The cells go straight from extraction to engineering, and they rely on the patient's own body to handle that final transformation on site.
It creates a highly scalable, just elegant manufacturing pipeline.
All right, let's do our second check-in here.
That sounds good.
So we've got the delivery sorted.
Right.
CAR T cells lack penetration, CAR-macrophages get lost in the liver. But monocytes naturally home strictly to the injured heart and are easier to manufacture.
Spot on.
And for you listening, how does it change your view of medicine to realize scientists aren't just inventing new chemical drugs anymore, but literally hijacking the GPS systems of your body's own first responders?
It's a total paradigm shift, but successfully delivering an engineered monocyte to the heart to eat the scar tissue, that bulldozer phase is only addressing half of the pathology.
Right, because if you program an army of cells to aggressively chew up the biological concrete holding the damaged heart together, you are going to leave a massive structural void.
Exactly. The remaining thin layer of muscle could easily tear, causing a fatal rupture.
So destroying the scar isn't enough. You must simultaneously rebuild the muscle. We need a builder to follow the bulldozer.
And this introduces the small p in pCAR-Mo, which stands for pleiotropic.
Pleiotropic, let's define that. It means a single intervention or single gene producing multiple distinct downstream effects.
Right. So the CAR portion, the chimeric antigen receptor, is the bulldozer's engine. It has a targeting fragment that binds to the FAP protein, paired with an intracellular CD3-zeta signaling domain.
So when the receptor grabs FAP on a scar cell, that CD3-zeta domain triggers the monocyte's internal machinery to initiate phagocytosis. It tells it to eat the target.
Exactly. But attached to this genetic payload is the builder, a protein called Agrin.
Agrin. Now, Agrin is naturally found in the extracellular matrix of neonatal hearts, right? Hearts that are still growing right after birth.
Yes, it's a fundamental growth factor.
But wait, if Agrin is a growth factor, a signal that tells cells to multiply, injecting it into an active biological war zone full of aggressive scar cells seems like a huge risk.
It does sound counterintuitive.
Like, why wouldn't the myofibroblast just hijack that Agrin signal to multiply even faster and build a bigger scar?
That is the critical mechanistic question. The brilliance of Agrin lies in its highly divergent, context-dependent signaling pathways.
Okay, break that down for us.
Basically, fibroblasts and cardiomyocytes possess completely different surface receptor complexes. Their internal wiring is different.
So they interpret the exact same Agrin molecule in opposite ways.
Precisely. When Agrin binds to a myofibroblast, it does not activate growth. Instead, it triggers biochemical stress pathways that induce senescence.
Oh. It forces the scar cells to artificially age and paralyzes their ability to divide.
Exactly. The fibroblast hits a biological wall and just ceases production.
That's incredible.
It is. But simultaneously, when that same Agrin molecule binds to specific glycoprotein complexes like dystroglycan on the surface of an adult cardiomyocyte, it unlocks a completely different door.
What happened?
It activates the ERK and YAP signaling pathways. In mature heart muscle, these pathways act as the molecular ignition switches that force a terminally differentiated, non-dividing cell to re-enter the active cell cycle and begin multiplying.
Wow. So it functions as a highly localized smart drug. It hits the brakes on the pathological scar cells while hitting the gas on the regenerative muscle cells.
Yes. And the researchers didn't just theorize this. They tracked 3 distinct pleiotropic mechanisms driven by the Agrin payload.
Let's go through them. The first one is enhanced phagocytosis.
Right. Agrin physically upgrades the bulldozer. It modifies a structural protein inside the monocyte called F-actin.
Because phagocytosis requires a cell to dynamically stretch and deform its internal skeleton to swallow another cell, right?
Exactly. By remodeling that F-actin network, Agrin makes the monocyte hyperflexible. It allows it to engulf almost 6 times more FAP-positive cells than a standard monocyte.
6 times. And they validated this using live cell confocal microscopy.
They did it. They captured real-time footage of the engineered monocytes just aggressively clearing the fibroblasts.
That's the bulldozer. Now, the second mechanism is remuscularization, the actual regrowing of the heart.
Right.
But how do you definitively prove that an adult heart cell, which is notoriously stubborn and refuses to replicate, is actually dividing?
You look for the specific molecular exhaust of the cell cycle.
The exhaust, I like that. What were they looking for?
The researchers tracked a series of highly specific proteins. They stained for Ki-67, which only appears when a cell is actively moving through the replication cycle.
Okay.
And they looked for pH3. Think of pH3 as catching the cell with its bags packed.
Catching it with its bags packed.
Yeah, it indicates mitotic chromatin condensation, the exact moment the cell's DNA bundles up tightly right before it splits.
Oh, that makes sense.
And finally, they tracked ARC-wipe, a protein responsible for actually pulling the duplicated chromosomes apart.
And what did they find?
The expression of all 3 markers spiked dramatically. The YAP and ERK pathways triggered by Agrin were successfully forcing mature tissue to clone itself.
Unbelievable. But a massive influx of new, highly active muscle cells creates a new problem, doesn't it? Oxygen demand.
Yes, you can't grow thick new heart tissue without a blood supply to feed it.
Which brings us to the third mechanism, angiogenesis.
Right. The Agrin payload also interacts with endothelial cells. Those are the cells that form the lining of blood vessels.
And how do they test that?
In laboratory assays using human umbilical vein endothelial cells, or HUVECs, the presence of Agrin significantly boosted cell migration and tube formation.
So the therapy is actively orchestrating the weaving of a new vascular network, just to sustain the metabolic demands of the regenerating heart muscle.
Exactly. It's a complete package.
Okay, let's pause for our third summer. This is a dense one.
It's a lot of mechanisms for sure.
So the recap. The SARA receptor eats the scar. The Agrin payload boosts the eating process. Then Agrin simultaneously forces heart cells to multiply, and it grows new blood vessels to keep them alive.
The perfect dual-action bulldozer and builder.
So for you listening, think about this. It's rare to find a single tool that destroys the bad while nourishing the good. Where else in your life or work could you apply a dual-action bulldozer and builder approach?
It's a great mental model.
It really is. Now, proving this intricate choreography works in isolated cell cultures is one hurdle. Proving it works in a chaotic living mammal is entirely different.
Totally different. So to test the clinical viability, the team utilized a rigorous surgical model in mice called left anterior descending, or LAD, coronary artery ligation.
Okay, walk us through that. What does that actually mean?
The surgeons permanently tie off the primary artery feeding the left ventricle. They're deliberately inducing a massive, severe myocardial infarction.
So they are replicating the exact mechanical and cellular trauma of a major human heart attack.
Exactly. And following that injury, they injected the pCAR-Mos intravenously.
And the functional results of that injection are paradigm altering. To measure how well the heart was recovering its ability to pump, they tracked 2 things. LVEF, which is left ventricular ejection fraction, and LVFS, fractional shortening.
Those are the gold standard metrics for cardiac output.
And the mice treated with the pCAR-Mos showed profound, sustained improvements across both metrics, right?
He did. And when they examined the cross-sections of the heart tissue later, the area that should have been a dangerously thin, translucent scar was visibly thick with dense, newly formed muscle.
But wait, in a real clinical setting, a patient rarely receives gene therapy within hours of a heart attack. Did they test for delayed treatment?
Crucially, yes. They delayed the intravenous administration of pCAR-Mos to day 3 and even day 7 post-infarction.
Wow. Day 7 is right when those FAP-positive myofibroblasts are at their absolute peak aggressive state, right?
Exactly. They gave the scar tissue a massive head start. And even then, the therapy successfully halted the fibrotic expansion, reduced the ultimate infarct size, and significantly restored pumping function.
That is staggering. But anytime you introduce a living, multiplying therapy that aggressively alters tissue structure, the immediate question is off-target effects.
Always. Safety is paramount.
Right. How do we know the monocytes didn't just trigger a systemic immune crisis or accidentally start eating healthy tissue elsewhere in the mouse?
To answer this, the researchers returned to single-nucleus RNA sequencing, but on a massive scale.
How massive?
They sequenced the transcriptomes of 18,840 individual cell nuclei harvested directly from the treated hearts.
Over 18,000 cells.
Yeah. By utilizing microfluidic droplet technology, they were able to barcode and analyze the precise gene expression of every single cell in that cross-section.
And what did the data show?
It provided absolute molecular clarity. First, the specific profibrotic fibroblast subsets, which are categorized in the data as myo, F-psych, and FIR, they experienced a sheer clunk drop in population.
The bulldozer effectively cleared the concrete.
It completely cleared it. But the truly unprecedented detail hiding in that data was the cardiomyocyte count.
The muscle cells.
Yes. The total number of functional beating heart muscle cells in the pCAR-Mo treated group was an astonishing 4.6 times higher than the control group.
4.6 times higher. That's not just a marginal improvement.
No, seeing a 4.6-fold amplification of adult cardiomyocytes in vivo, it completely rewrites our understanding of cardiac regenerative limits.
And the RNA sequencing confirmed the upregulation of those specific cyclins and CDKs we talked about.
Exactly. The very gears of the cellular division engine were turning. The cells weren't just surviving the localized trauma, they were robustly multiplying.
Okay, but what about the safety profile outside the heart?
The sequencing and subsequent tissue analysis of the liver, lungs, spleen, and kidneys showed zero toxicity. Zero off-target tissue damage.
That's incredible given how aggressive the therapy is.
It is. The therapy remained remarkably confined to that FAP-positive microenvironment. In fact, by rapidly clearing the necrotic debris and the pathological scar cells, the therapy actively downregulated systemic inflammatory pathways.
Oh, so instead of causing a cytokine storm, it functioned as a precision cooling mechanism for the immune system.
Exactly. It calmed the whole system down by fixing the root problem.
Let's do our final check-in for the listeners.
Correct.
So in living mice, pCAR-Mos reversed heart failure, increased heart muscle cells by 4.6 times, caused zero systemic toxicity, and worked even when administered days after the heart attack.
It's a pretty flawless proof of concept.
It really is.
Yeah.
So for you listening, seeing 4.6 times more heart cells in an adult mammal is unprecedented. What's the most surprising or aha detail from the sequencing data for you?
It's a lot to process. But it's so important because it synthesizes why this matters. We aren't just treating the symptoms of a heart attack anymore. By utilizing the unique homing of monocytes and the dual action of CIR and Agrin, we are fundamentally remodeling the tissue microenvironment.
And it makes you wonder, as someone simply observing the velocity of this medical technology, if scientists can literally hijack your body's own ambulance system to clear rubble and regenerate an organ as complex as the heart, where else can this architecture be deployed?
That's the billion-dollar question. Because fibrosis, the pathological, relentless scarring of tissue, is not unique to cardiology. It is the terminal mechanism for dozens of diseases.
Right. So the foundational architecture of this therapy is modular, isn't it?
Exactly. The monocyte chassis provides a universal delivery vehicle to any site of severe inflammation. If you swap out the anti-FAP receptor for a different target and carefully tune the regenerative payload, this exact eat and rebuild mechanism could theoretically be adapted for almost anything.
Like liver cirrhosis, reversing end-stage hepatic failure.
Yes. Or it could be targeted at renal fibrosis in the kidneys or deployed into the lungs to clear out pulmonary fibrosis. The heart was simply the ultimate proving ground for the delivery mechanics.
Wow. We started this deep dive looking at a fractured biological repair mechanism, right? Where the body's frantic attempt to heal a wounded heart results in a rigid, lethal scar.
And for the entire history of modern cardiology, that scar was a permanent, irreversible endpoint.
But by mapping the exact chemical distress signals of the heart, hijacking the natural GPS of circulating monocytes, and arming them with this incredibly precise biological toolkit, Wu and his team have demonstrated that the damage can actually be unwound.
We now have the blueprints to program a patient's own immune cells into autonomous regenerative engineers.
Which leaves us with a final, profound question to ponder about the trajectory of medicine.
Yeah. If we can systematically clear pathological damage and orchestrate tissue rebirth at the cellular level, will scar tissue soon shift from a permanent condition to an entirely curable disease?
It's an amazing thought to end on. Thank you for joining us on this deep dive into the expanding frontiers of cellular engineering. Keep questioning the mechanisms of the world around you, and we'll catch you on the next deep dive.